I work in a cancer discovery unit, and our mission is to discover novel anti-cancer therapeutics. We focus exclusively on small molecules, and we're probably the large non-profit organization in Europe working in drug discovery. Despite this, we are actually quite successful. Abiraterone has been registered 2 years ago for advanced prostate cancer. This is an HSP90 inhibitor, which is currently with Novartis in development. This is a PI3K inhibitor that I will spend some time discussing today, which we developed and is currently with Genentech. These are PKB inhibitors that are in clinical development. One is with AstraZeneca, and this one is still with us. We're taking it to phase 2. We have some compounds in preclinical development, and we have a preclinical portfolio of 8 targets. So what does it take to make a drug? Well, you have actually to select the right target. You have to identify high-quality hit matter, so that means good assay and also good chemistry. But also, you have to identify good biomarkers and actually make sure you have the best development candidate that you're going to put in the clinic. And finally, you actually have to design the right clinical trial to develop the agent. So a large part of what we do in optimization is actually to get this golden triangle here, find the right compound levels, the right schedule, find the right target modulation, and that should result in the right efficacy. And in cancer, we have animals that bear tumors, so it's really easy to actually measure the target modulation in the tumor sample. By the time we get to phase 1 clinical trials, and for those of you who are not in oncology, phase 1 clinical trials are not done in human healthy volunteers. They're done in cancer patients, and normally they've gone through many courses of therapy. So you have to do the same thing, and we've gotten much better at predicting the PK. But you also have to monitor the fact that you've got your right compound levels, but you also have to show that you're modulating the target. And you cannot get many biopsies in a cancer patient. You're lucky if you get a pre-treatment biopsy and a post-treatment biopsy. And these are for patients who've got accessible tumors. So if you have a melanoma, it's very easy. If you have circulating a blood disease, it's quite easy. But if you actually have prostate cancer or liver cancer, it becomes really risky and difficult. Therefore, we actually need to have circulating biomarkers. And this is what led me to metabolomics, because I thought, well, if we can actually find something in the blood, then obviously we will be able to follow it up across time and in multiple samples. So the PI3K pathway, well, all the people in oncology, I mean, there are many of these inhibitors in the clinic at the moment. It's a very activated pathway in oncology. It's activated at the level of growth factor receptors. This is P110 alpha, which is one of the isoenzymes. And it's a lipid kinase. It phosphorylate phosphatidylinositol biphosphates into triphosphates. These are lipid second messengers. And what they do is they actually recruit AKT to the membrane. AKT gets phosphorylated, and it results in a cascade of events that actually end up making cell cycle progress and proliferate, affect glucose metabolism, survival, motility, invasion, and protein synthesis. Now what's interesting, and there are many ways to inhibit these pathways. So I've told you we've got some PI3K inhibitors, got AKT inhibitors. And in the interest of what it will follow, the negative regulator of the pathway, the phosphatase PTEN, is actually null in many human cancers. This is mutated in many cancers. This is amplified or mutated. There are activation upstream, many, many ways to activate this pathway. So this is the compound that I'm going to talk to you about, GDC0941. The name that Genentech have given it is unpronounceable. Try. So this is our lead optimization. We started with this compound, which is a tricyclic core structure. very insoluble, very highly cleared by virtue of glucuronidation of this phenol ring. And we basically could see that you could actually knock out one of the rings, get this bicyclic compound still very active on class 1 inhibitors, or class 1 enzymes, sorry, but still really glucuronized. If you replace this phenol by an indazole phenol isosteres, you get still a very potent compound, soluble with good plasma clearance. So all these compounds have got very good anti-proliferative in panel of cancer cell lines with loads of different genetic abnormalities. In vivo, this is a human glioblastoma xenograft, so grown under the skin of nude mice who are immunocompromised. And you can see the tumor sort of growing in the control group, and the growth is inhibited in a dose-dependent manner here. These are the final tumor weights. You can see that you can completely obliterate the tumor growth with even a bit of regression here in some of these groups. And this is the biomarker in tumor, control. And then you can see a dose-dependent inhibition of the biomarkers as AKT phosphorylation in these animals. So what we decided to do is to say, can we actually identify a metabolomics marker in the plasma by LCMS in preclinical models? And we did. And then we thought, well, this compound was in phase 1 clinical trials in our institution. Can we follow up these markers in the phase 1 clinical trial of GDC 0941? The preclinical studies design was as followed. So we used genetically engineered mouse model, and they were PTEN knockout versus wild-type littermates. So I told you PTEN is this negative regulator of the pathway. And then we used some xenografts. Some of them had no tumors, and some of them were tumor-bearing. 2 different tumor types, both are PTEN null. One is a prostate, the other one is the glioblastoma model for which I showed you the previous slide. So we either took control animals treated with vehicle, or animals treated with a GDC 0941 at various doses. So my lab is a pharmacokinetics lab, so we have very geared up towards reverse phase chromatography. So we didn't do the whole extent of the metabolome like many of you do. I only looked at reverse phase chromatography in positive and negative ionization mode. So the non-targeted metabolomics that we used actually led to a large number of metabolites, and we could identify 30% of them. Like many others, many of those remain unsolved. And some of them, of course, we wanted to take the study into clinical trials. Some of these metabolites were uncomfortably close to the solvent front, like all the amino acids. And I thought, well, I'm not taking this to the clinic. This isn't good enough. I was a little bit worried, and I thought, wouldn't it be nice to actually... So we started quantitating each and every metabolite and, developing other methods. And then we saw that many, many of them were actually in the Biocrates absolute P110 180 kit. So rather than reinventing the wheel, we went and confirmed our data with the kit, and then took the kit to the clinical trial. So these are the mice that have got activated tumors, and they show a modified plasma metabolome if you compare them to normal control animals. So these are the genetically engineered models, and you can see that most of these metabolites are actually decreased. And then same, the PC3 tumors versus the non-control animals, and the glioblastoma tumor-bearing animals versus the control. And overall, what we saw is a decrease in branched-chain amino acid, aromatic amino acids, acyl carnitine, lysophosphatidylcholine. So all the amino acids are here, short-chain carnitine, long-chain carnitines, lysophosphatidylcholines, and phosphatidylcholines. So can we do anything if we treat with the inhibitor? So these are the animals that I've just shown you. This is the metabotype of these animals. And if you actually treat animals that bear these tumors, the U87 or not PC3 with GDC0941, you can see that all these are gray, and now all these are turning to red. You basically reverse what you've observed in those animals. And if you actually treat with the inhibitor a non-tumor bearing animal, you actually don't see the same effect, maybe slightly at the early time points here, 1 and 4 hours, but by and large, you don't see the same metabotype. And I'd like to say this isn't an end of study, this is just a single dose. So it's not like the tumors disappeared, so the metabotype, you're actually reversing these effects. So if you take a standard of care, carmustine is a BCNU, it's an alkylating agent, and you treat animals bearing this U87 with the carmustine, you actually don't quite recapitulate the same metabotype. So obviously a cytotoxic does something different. So what happened in the clinical study? So this is the clinical design. So as I said to you, these are patients that have had seriously been pre-treated prior to therapy. Normally, these are people who have no therapeutic hope. They've been through every conventional therapy, yet they have a good performance status. And this is a classical sort of mixture of patients, varied malignancies here, 41 patients. So the patients received, and I have to say, because this was one of the first PI3 kinase inhibitor to reach the clinic, everybody was very worried about toxicity. So the design was to treat for 1 day, then wait for a week and start treatment daily over all the cycle 1, and then resume and start cycle 2. So we sampled on day 1, 2, 8, and 24-hour post-dose, and then on day 8 again, and then on day 15, right in the middle of the treatment. So what I'm showing you here is the PC3, the sort of PTEN null animals. This is the results in the xenograft experiments, just one of them. And this is in the patient population. So this is the first dose level, 40 to 80 milligrams, first doses levels. And at those dose levels, we didn't actually see any biomarker modulation as phospho-AKT inhibition. And we actually didn't see, we saw very little modulation of the selected biomarkers that we were looking at. In contrast, at doses where phospho-AKT was actually modulated, we could see the same effect on the amino acids that we'd observed in the preclinical models. We could also see the same effect on the short-chain carnitine, but the long-chain carnitines actually behaved in the opposite way. In fact, they behaved more like the non-tumor bearing animals. And the lyso-PCs were again showing a similar pattern to what we'd identified preclinically. So these are just to show you that the effects were dose dependent. So if we take, for example, valine here, you can see that at 2 hours, we've got no effect at the early doses and then a dose-dependent effect here at 6 hours and 24 hours. And if we take the same marker here on day 1, that's what I've just shown you. This is just a high dose, the highest cohort. And then the effects were recapitulated. The day 8 baseline was back to normal. So you can see that the sort of background, the baseline levels are actually quite reproducible. And you can see that by day 15, you actually see that same effect again. So the dose-dependent effect of GDC941 are restored by day 8 when you stop treatment and recapitulated on day 15. So what does it all mean? Well, all the metabolites that I've shown you, the branched-chain amino acids, the aromatic amino acids, the short-chain carnitines, and the lyso-PCs are consistent with insulin resistance associated with PI3 kinase inhibition. And in this trial, we were able to also look at insulin in the plasma of these patients And you can see that compared to baseline, we had a shift. And it's a bit difficult to say, because at the low dose level, we had no effect. But then we started to see a shift up to 2 to 3-fold variation in insulin levels that were mirrored by the glucose levels in these patients. And of course, we know that insulin signals through PI3 kinase, and there is a feedback mechanism via P70S6 that regulates this whole pathway. So everything we know it fits with the metabolite that we have observed here. I'd like to say that we've worked with other signal transduction inhibitors, and I won't show you all the data today, but we've worked with a MEK inhibitor in different tumor types, and basically there is a signature of about, again, 18 to 20 metabolites. Only 3 of those are common with the PI3 kinase pathway. So we believe that we're onto something real here. In conclusion, we have identified distinct plasma metabolites consistently altered through preclinical models. In the phase 1 clinical study, we were able to show that there is a time and dose-dependent modulation of the biomarkers, and these findings provide a link between PI3 kinase pathway modulation and amino acid acyl carnitine and lysophospholipid. They're consistent with the insulin-resistant phenotype associated with PI3 kinase inhibition. I'd like to thank you for listening, and also thank all the people who've contributed to this work. The non-targeted analysis was done by Ruby Panda in my lab. The targeted was done by Juan Ang. I'd like to thank all the people in the clinical study, Genentech for allowing us to present the work, the in vivo team, and the patients and their families. And you for listening. Thank you.
How difficult was it for you guys to qualify these kits and so forth for the clinical application?
Actually, it was really easy because we had some controls that we measured, that we knew the sort of levels of, and we overspiked a couple of samples. And basically the kit provides its own quality controls as well, and you can also run control plasma that you know well throughout those studies. So the advantage is it's already qualified in a way, and it's a lot more reliable than anything you would do with non-targeted analysis.